I'm happy to introduce and welcome everyone to the Journal Club, obviously, today. Darrell Eacret is a neuroscience graduate student in my lab, and he has been studying actually this bidirectional relationship between opioids and sleep disturbances. And I think along with sleep, we often think about circadian rhythms. And so I think this particular paper was of interest to think about sort of how we go about measuring things and studying them with the biological clock in mind. So without further ado, Darrell, I guess you can take it away.
All right, thank you, Julie. And
Raise
people
your
see
hand.
us just fine
There you go.
and hear me all right? Yep.
We can.
Okay.
Indeed.
Yes, so the paper I'm talking about today is called Seasonal Variation in the Brain mu-Opioid Receptor Availability. It's from Sun et al. From a group in Turku, Finland, and was just published in the Journal of Neuroscience earlier this year. And as Julie alluded to, I liked this paper because it relates to my interest in sleep and circadian rhythms and the opioid system in the brain, as well as I thought they did a good job having a parallel line of research with humans and rats. And I thought that was interesting how they linked the two and had one retrospective analysis and one causal analysis. I thought that was an interesting part of the paper. So to give some background about kind of the relationship between seasons and opioids, it's widely acknowledged that seasonal rhythms affect mood. So seasonal affective disorder is a disorder where negative affect is worst in the winter months and best in the summer months. There's some evidence that the opioid system relates to mood too. So this convergence on the question of whether the opioid system and the convergence of seasons can contribute to mood and social effects. Additionally, this lab is from Finland, as I mentioned, Turku, Finland. And so this is probably more salient to them than it is to us because their daily rhythms of sunlight go from, or daylight go from about 6 hours in the winter months of December and January, all the way up to 19 hours in the summer months of June and July. So they have more extreme changes in day length than we do here in Philadelphia. So this is more of a salient question to them, but interesting in terms of seasonal changes in day length and how that affects the opioid system and potentially our mood and social behavior. Further interactions between the opioid system and seasons. Lower opioid metabolites are found in the wastewater in the summer months. Opioid overdose deaths are also lowest in the summer, so this potentially converges to make us think that potentially there's lower opioid use in general in the summer and more in the winter months. Opioid-related suicide attempts peak in the spring and fall, and suicide attempts in general peak in the spring, regardless of the geographic location. And then there have been a few studies in animals showing seasonal differences in mu-opioid receptor expression in the brain across different seasons. But this is still a bit of a novel field, and again was what I thought was an interesting part of the paper, how they decided to look at this question. So they did PET scans in both humans and rats. And for people who are not as familiar with PET scans, sort of like me, how it works is a specially designed radioactive molecule is used for a specific function. So in this case, they used 11C carfentanil, which is based on the synthetic opioid agonist fentanyl and has radioactivity and can be traced in the PET scan. So this molecule is injected into the bloodstream, it gets absorbed by the brain, and a radioactive atom on this tracer eventually loses its activity and will give off a subatomic particle called a positron. This positron will eventually hit an electron, and the positive and negative forces combine and destroy each other, and the energy is released as gamma rays. And this circular PET scanner can detect the gamma rays from two opposing sides and can calculate where in the body that tracer must be thousands of times per second. and fancy computer processing can reveal the 3D location of the target in the brain, and you can end up with cool images like this that we'll see later on in the paper from the PET scan. So this is the human version. They also did it in mice. The difference is it's easier to do repeated scans, with rats. It's easier to do repeated scans with rats. It's not necessarily safe or logistically possible to do so in humans. So they, what they did in humans is use this huge database of PET scans that they had from the Turku PET Center in Finland, and they did a retrospective analysis of human subjects from many PET scans that were done with 11C carfentanil at different points of the year. So they have this AIVO database where PET scans were done with all types of different tracers, but they can focus on the ones done with 11C carfentanil here. And they also have certain information about the subjects, such as whether they have any neurological or psychological disorders, their age, their sex, and other data like that. So again, they focus just on PET scans done with 11C carfentanil to target mu-Opioid receptors. Again, 11C carfentanil is a radioligand based on fentanyl, and it's fairly specific for mu-Opioid receptors. So what they're getting at is availability of mu-Opioid receptors from 11C carfentanil. And there were 471 PET scans available from this database that were done with 11C carfentanil. The researchers wanted to make sure that the individuals from the scans had no neurological or psychological disorders. And so they ended up, out of 471, they ended up with 204 from the years 2003 to 2018, 132 males and 72 females with an average age of about 32. And this was done in 11 different studies and 5 different PET scanners. And they had a computer model that controlled for the effects of different scanners. They also used a program in R called Suncalc that, based on the day the PET scan was taken, they can figure out how long the day length was on that day. And they plotted a histogram here in figure 1A of the different lengths of daylight that existed on the day these subjects were in the PET scan. So the range here is at the low edge, we have 7 hours of daylight, and at the high end, we have 23 hours of daylight. So that's almost a full day of sunlight. So what they found was that there are day-length sensitive brain regions for mu-Opioid receptor availability. So in A, first, what we're looking at is just the distribution of mu-Opioid receptors, and they use BPND as their units. BPND is the ratio of specific binding to non-displaceable binding in the tissue. So the higher levels and the warmer colors indicate higher mu-Opioid receptor availability, meaning higher tracer binding to the mu-Opioid receptors. And here we can see the higher levels kind of around the thalamus and lower levels at the edges of the cortex and near the ventricles. In B, we see the profile of day length. So how the researchers here defined day length was the solar day, so when the sun is above the horizon, plus civil twilight, which is when the sun is within 6 degrees below the horizon before sunrise and after sunset. The civil twilight adds a little bit of day length to the time that the sun is actually above the horizon. And again, they have a range from about 7 hours of day to about 23 hours of day, including civil twilight. And what they did in C was look at whether there are brain regions that vary according to the day length. And they did find that there is a cluster of brain regions that are sensitive to day length with respect to mu-Opioid receptor availability. They found a cluster sensitive to day length that encompasses the superior frontal, medial frontal, middle temporal, and superior temporal gyrus or gyri. as well as the insula and orbital frontal cortex. And together, they say that these are certain regions that do have effects on mood and emotional and social status. And so these regions do change with respect to day length based on their retrospective analysis. Furthermore, within this cluster of regions that were activated with respect to changes in day length, they found that BPND, or mu-opioid receptor availability, was highest at 15 to 19 hours. And the receptor availability actually followed this inverted U pattern. So at the short and long amount of day length, there's lower mu-opioid receptor availability than the intermediate lengths. And the mu-opioid receptor availability peaked at about 15 to 19 hours of daylight. So it's interesting that it's not a linear relationship between light and mu-opioid receptor availability. It's more of an inverted U pattern. So they also plotted the BPND, so the mu-opioid receptor availability ratio compared to the different seasons. So again, spring had the highest BPND, which follows with this peak at the intermediate day length of about 15 to 19 hours. So summer, fall, and winter, they appear pretty similar, and then we see a higher BPND in the spring. And in B, we are shown the plotted day length. So summer obviously has the highest day length, spring is intermediate, and again, this is where there's the highest levels of mu-opioid receptor availability. So again, that human study was thorough with a good amount of samples. They had 200 and an N of 200, but again, those samples were only done one time. It was a retrospective analysis, and they didn't have other information about the humans, such as were they outside during that day, what was the temperature like, things like that. So in addition to the human data, they wanted to bolster it with a causal study with rats. So they did repeated dynamic PET imaging in rats. They had 6 male and 3 female Sprague-Dawley rats, as well as 2 male and 1 female controls. So the experimental rats had a changing day length. So the colony room that they were housed in changed the lights on period from 18 to 19 hours at the higher levels, to 12 at the moderate levels, and about 6 at the low levels. And these rats were imaged 4 different times throughout changing levels of day length, while the controls were maintained on a standard 12-hour light cycle. And this was a speeded cycle that was completed in 3 months. So not a full year, but they were able to change the day length 4 different times and repeatedly PET scan the same rats over time. And in 4A, again, they used 11C carfentanil, so the same radioactive tracer that they used in humans, they used in the rats here, and found the activity peaks at about 15 minutes after injection in the striatum, thalamus, and neocortex. The cerebellum was used as the reference region because the cerebellum has low to no mu-opioid receptor binding, so that this is a good negative control, whereas these other regions show higher binding that goes away over time after about an hour. In C, they did ex vivo gamma counting. So after the last PET scan, the animals were sacrificed and they counted the particles in the brain and saw high levels indicating higher binding in the neocortex, striatum, and thalamus compared to the cerebellum, where there aren't mu-opioid receptors. And what's really interesting and impressive is they found a very similar inverted U pattern of BPND, or mu-opioid receptor availability, in the neocortex and striatum and thalamus as they saw in the human study where in the activated cluster responding to day length, there was this inverted U pattern. So they saw the same in the same rats in PET scanned over time. This inverted U pattern held up in these 3 brain regions. And additionally, day length was a significant predictor of mu-opioid receptor availability. So the summary is that seasonal variation in day length influences in vivo brain mu-opioid receptor availability in both humans and rats. The highest mu-opioid receptor binding occurred in intermediate day length, and brain regions sensitive to variation in day length with respect to mu-opioid receptor availability occurred in a cluster that encompasses the mood and social related brain areas. Some strengths and weaknesses of the paper. I thought it was really impressive how they found consistent results across humans and rats. That's often hard to do. Some of the things you see in rodents don't necessarily appear in humans and vice versa. This was a strong, consistent pattern across both of them. They had both a retrospective correlation in humans as well as causal evidence in rats where they could pretty directly control the day length and show that manipulation was the result and the reason for the changes in mu-opioid receptor availability in those brain regions. This is the first study to show in vivo evidence of seasonal variation in the mu-opioid receptor system in humans. So again, I thought a nice, interesting hypothesis, the first to do it, a clever way of using the database, the big PET scan database that they had in Finland. And then a few limitations that I thought, again, going back to that histogram in figure 1A, they had few scans with a long day length. So most of the subjects were done at about 7 to 15 hours. And on the other end of the tail, they didn't have as high of ends. Potentially not a diverse population, all coming from Finland. So I'd want to see if this holds in the United States across different races and socioeconomic statuses and different types of and ages. Again, they lacked information about some of these participants. So when these authors are claiming that if the seasonal changes in day length with respect to the mu-opioid receptor could contribute to mood and social behavior, it would have been really nice to have a questionnaire regarding mood and social patterns and data from when that PET scan was taken. Unfortunately, they couldn't because they're just grabbing the PET scans from what existed previously and didn't do a full study dedicated to this question. Also, they didn't show stress or weight data in rats, so they collected body weight data, they collected serum corticosterone, and actually had some interesting effects. They had, in the experimental group that had changes in day length, they showed lower weight gain compared to the control day length and higher corticosterone in the experimental group. They also had corticosterone was, the levels of corticosterone was associated with mu-opioid receptor binding in the striatum, but this is just left as kind of random sentences in the text, whereas I think they should have included this in graphs in the figures. Especially because in the discussion they mention potentially stress as a reason for the inverted U rather than a linear relationship with day length. So I would have liked to have that be more of a feature, especially because they're using the rat study as a causal study where the independent variable is changes in day length. So I would have liked to have them feature this more, especially when they have interesting results. And I have a few discussion questions, so I ended a little quickly, so please join in with some of these. I mentioned in the introduction that opioid overdose deaths and metabolites were lowest in the summer, so when at the high end of the inverted U. So past the point of optimal mu-opioid receptor binding. So why isn't there a linear relationship? Why not just more light equals more mu-opioid receptor expression? Is this because the longer day length contributes to higher corticosterone levels or cortisol levels in humans? Does this kind of negatively inhibit or inversely associate with the mu-opioid receptor system? Is there a physical connection between the retinal ganglion cells, and the central mu-opioid receptor system? They mention a little about the retinohypothalamic tract. So how does information about the light coming from the retina actually get to the brain or the hypothalamus and affect the mu-opioid receptors directly? This wasn't, I don't think there's much information about this, but that's potentially an interesting basic science area of research. As Anna Rose mentioned at the beginning, is this study convincing enough that any PET study done with 11C carfentanil, should we have another factor that we have to associate or that we have to correct for? So if you have studies looking at mu-opioid receptor availability, one's done in July, one's done in January, can we directly compare these? Can we use those same scans in one study? Or does there have to be a day length correction taken into account in the future? How does sleep and circadian rhythms influence the central mu-opioid receptor system? So there is some evidence for changes in sleep across the seasons. Could this contribute to changes in cortisol, changes in the mu-opioid receptor system? Again, they don't have this information in the human subjects that they collected. So is this potentially another factor? If they're doing a new study, do they have to take this into account? And then can you do something like light therapy or light manipulation? Could that treat opioid use disorder? Or on the flip side, could mu-opioid receptors be a target for seasonal affective disorder? So please, anyone jump in with further questions or answers to these discussion questions, and thanks for your attention.
Thanks so much, Darrell. That was a terrific overview, and I think the article was really intriguing because of its intentional combination of these preclinical data and of course having the large clinical data set from which to draw. I mean, it took years and years and years to accumulate those PET scans, and so everyone knows a PET cohort is not a fast thing to acquire. So my guess is people have to end up sort of taking day length into account rather than waiting till the same time next year to scan, because things are so difficult anyway. But I think your first question is an important one. I'd like to hear people weigh in on that. What are the thoughts about mechanistically what might underlie the inverted U relationship? If you want to talk a little bit more about that, or get someone else to say something.
Yeah, that's what I was hoping that some of the carfentanil folks that are on the call could speak to that, and also, is this can this be ascribed to differences in scanners, or were there a number of people in enough different scanners that could that really contributed to the inverted U? And it looks like Jacob is going to talk about that.
There's a couple issues with both populations. So I would say PET scanners have changed a lot since between 2003 and 2018, and 5 different PET scanners, and they're in my division when there's a discussion of, say, clinical trials, when you're trying to look at patients at different sites, and I think Robert's on the call too, where he has done the work in terms of kind of cross-calibrating these scanners where there's a significant amount of variation between scanners. And so not just you have 5 scanners, so that could be a problem. I'm not sure. I can't speak to the program they use to compare the scanners as well. I'm not familiar with it. But that can be, I mean, they try to reconstruct them the same way. And I assume their synthesis of the carfentanil was the same, throughout all these different these multiple studies. But that's one aspect. One thing that they reference in the paper, which I think you should mention, which is something that's important for PET imaging if you're using a tracer, is they reference their own test-retest variability paper, which they found less than 6% test-retest when they did. So I think that they did that many years ago, but I think at least in 2009 it spoke to the rigor, and I think that supports what they were doing. However, I do find their animal prospective validation flawed, in that Sprague-Dawley rats are different vision, and they're also nocturnal. So how are we validating a photosensitive kind of influence on mu-Opioid receptors with an animal that spends most of its awake time in the dark? I don't understand how you can compare the two species that way. That's my biggest issue with the prospective examination. If they had, non-human primates or something else, okay, but I don't think this validation holds the same water because of the species difference in the behavior. That's what I worry about from this paper.
This is Robert. So one of the first things I look at is, first off, very interesting study, and I'm very glad we chose to look at this paper because it's kind of a fascinating topic. But in terms of practical application to our studies here and to our P30, if you'll note from the paper that the change in daylight hours in the area where they study it was like almost 16 hours, 15.5 hours, 15 and a half hours, whereas in the Philadelphia area, it's only a change of about 5 hours. So it's a much less dramatic daylight period of time. So I would be somewhat skeptical that we're going to be able to, that amount of a change is going to be enough to even make a blip of difference in what we're doing. Now, if we were further north, then maybe it'd be more of a concern. I'm not too concerned about here in Philadelphia unless we're flying in people from Alaska or other locations. I guess the only thing is that this is just a general comment, it's not so much on the PET. I'm just curious why the MRR was like the worst for these patients seemed to be in the spring. Does anyone care to comment why it would be in the spring? It seemed to correlate with suicides.
Yeah,
we were trying.
That was curious. I mean, I would love for people to comment on the mechanism in terms of that. They did nod at the end to the fact that obviously, it's binding potential, and that basically you're taking in a baseline scan like this, not trying to stir up the endogenous opioid system. You're taking that as sort of an index of the density or concentration of the receptors, even though this is a competition ligand in some sense, so that if you stir up a lot of endogenous opioids, they should be competing with the tracer. So, yeah, I'd love somebody to comment on that. But good point about the fact that Philadelphia is not as far north as Finland.
Yes.
And I would say one thing, and just thought about all the variability in the different protocols and the different platforms, is that all those things would essentially throw variability in the data and would actually go against you getting some kind of systematic pattern. So the fact that they got a pattern despite those, unless somehow those things were systematically in some way we don't know creating a pattern, seems like, yeah, there's could be potential differences, platforms and so forth, but the fact that they managed to have a pattern that emerged even with those differences taken into account, I think is pretty intriguing. I don't know. I don't know how common it is to be able to push a day-night cycle with animals, and Jake's point's an interesting one in terms of the nocturnal versus more diurnal animals like the primates. So, those are good points too.
The thing that has me a little bit concerned, and it's on, if you keep this slide here, it proves the point, is the amount of between subject variability. The test-retest variability is only 6%, and the between subject variability is 100%, right? It's going from 0.6 to 1.2. what I would have liked to have seen, and what I would have requested if I was reviewing his papers, I would have liked to have seen the binding potential per year, and how has it changed for the different scanners, and how has it changed from the different chemistry modules that were used in the synthesis of carfentanil, because it's a very large between subject variability. And looking at that, I would argue that there's really no difference between spring, summer, fall, and winter. I mean, it's spring is a little bit higher binding potential, but is it really significant? I mean, it's just way too much variability there. It does raise the point from our hands, though, is we perhaps need to pay attention to this as we begin our carfentanil studies, and we need to make sure that we have appropriate age groups, apparently not only, or appropriate controls, not only match for age, but match for age in time in the season in which they were scanned, right?
Or that we should at least take note of that. I mean, and then maybe have to parse it out statistically.
So I
mean,
agree
Robert,
with,
what do you think of that between subject variability on that slide that was just up there? It's really high.
Let's see. Which figure are you pointing to?
A right here.
Yeah, A.
Yeah. Yeah, well, the other thing that makes me a little bit skeptical, and I know this just because I've looked at too many studies, is that usually when you're the very best center and you're doing something that you've done a lot of times, it's pretty hard to get below 10% on any parameter. 6% is phenomenal. I'm kind of shocked it was actually that good. That seems a little better than I ever see reported for a repeatability. So that makes me a little skeptical.
Yeah, Rich Carson just published their UCBJ, and it's like 15%. And I
I'd
figured
believe that. I'd believe 10%, around 10%.
Is tight.
And then if you're doing multi-center, you're getting closer to 20% or higher. So
before we go off the rails, I think Darrell did a great job presenting, and that's kind of like that is in the detail. It's one of the devils in the details of the PET, and that's something that we in the PET world get concerned with, the test results.
But that's exactly why it's fun to have this as a discussion article, right? Is that there's the article that we all read, and then there's the inside scoop from you guys that are working with these data every day and know about things like the test-retest and what looks, very good, and then too good even perhaps for Finland. Who knows?
Well, if you want to get into the details, this is getting a little more in trees than I sometimes would go perhaps in this audience, but they use a simplified reference tissue model for doing their quantification, which is okay in and of itself. But what I'm guessing they did is they probably just had these values available in a catalog and they didn't actually model it from scratch. If I was modeling this from scratch and I knew that all I cared about was a binding potential ND, I wouldn't use the SRTM. I would use the Logan reference model. And it's just a more robust test. It's harder to get that wrong. You need less expertise. So I'm kind of surprised they used a little bit slightly more complicated model unless the data was already available to them. It just points to me they probably took this from a database, but I can't know that for sure. But that's nitpicky. I mean, SRTM in a lot of contexts would be fine.
Yeah.
I was going to say there's one other thing in the human data that they did not take an account of that's near and dear to someone's heart on this call that we should mention. And of course I'm talking about Julie. And of course I'm talking about the OPRM1 SNP, which has shown binding potential variability in human subjects. And so with it, that's another factor we know alters overall. And so we can't just ignore that obviously they did not do genetics on these 204 individuals.
right. And, the likelihood in this, European population of about 25%, given, 250, the likelihood of there being some G allele carriers is probably pretty high. But my question is, so this idea that receptor regulation has a circadian periodicity, I mean, that's kind of been around for a long time, right? People thinking of depression and a lot of disorders as biological rhythm disorders. And I guess the idea of some of the opiate receptors. I mean, typically people looked at serotonin receptors and the adrenergic receptors, but I don't think anyone's really looked at these, the various opioid receptors. And so while I agree that, it's unlikely for us to worry about, these time differences, I think the benefit of doing this in Finland is having these extremes and being able to then assess, 18 hours of daylight versus 2 hours of daylight. And so this kind of, to me, says, well, this can happen, and depending on the biology of how it happens is kind of intriguing to me. In terms of, and Darrell and I were talking about, the regulation at the mRNA level of opiate receptors. There's estrogen receptor regulation, but there's nothing much about any of these clock genes. There's nothing about like glucocorticoid receptor, regulation. So how is this happening? How is light, like you said, not only getting to the hypothalamus, but regulating, the rest of, these areas in the brain?
Yeah, and it's a good point that Finland, because it has the extremes, is sort of a natural place to look, right, for the things in extremis and then see what's there as a principle that may be, important therapeutically or understanding-wise in terms of vulnerabilities that are in, less dramatic form other places and other spaces. But mechanistically, I would be interested in what people think about in terms of basically why, how.
Yeah, there's some evidence about like retina to raphe and retina to locus coeruleus with serotonin and norepinephrine, these physical circuits in mice between the retina and mood-related neurotransmission, but I haven't seen anything with regards to the opioid system and how directly information from the retina gets to mu-Opioid receptors or other opioid targets.
Right. Seems, as they say, this is an area for further experimentation, but it's actually, but it's an intriguing one, and this was very provocative, I thought, even with the cautionaries that we've had from the technical folks.
So can I ask a somewhat related, but maybe not? How easy is it to tell if carfentanil is on neurons versus, say, immune cells and microglia in the brain? Can you distinguish that with PET scanning?
No.
No.
We can't. Well, it'd have to be some kind of dual tracer cell type, but I think that would more lend itself to some kind of microscopy IHC going on. That would not. We can't do cell type brain.
Okay, thanks. I kind of guessed that, but I was just hoping maybe something's come up since then.
There's a secret way that Robert's figured out. No.
We don't have that special sauce yet. We're working on it.
No. Yeah, we can just see radiation, so it's like.
Well, I mean, that's an interesting question, Julie. I'm debating whether or not to pull up a slide of our first non-human primate study we did with carbon-11 carfentanil last week. I just had the images sent to me a little while ago, but we do see a lot of, the advantage of the PenPet Explorer is we see the whole body imaging, and we do see high uptake of carbon-11 carfentanil in organs that are known to have immune function, like the spleen, with high uptake. So I think there are certainly, there is a possibility these receptors, I guess they are on the immune cells, right? And
Yeah.
It would be impossible to differentiate that in the brain because you're just getting a signal and you'd have to have a separate tracer to measure immune function to try to correlate with the carfentanil function. But we can look at carfentanil uptake in other regions of the body known for immune function, like the spleen. We even see high uptake in the bone marrow, which is something that caught me off guard when I was looking at the images. So I think there are ways that we might be able to study that on the Explorer that we can't measure in the brain, but we can look at the effect of these things on immune function outside the brain.
Yeah,
right.
I
Bob,
think that's useful.
I was going to suggest something that, I kind of was playing with the nicotine study where we see also the uptake in the marrow, which was when I give a nicotine challenge, we don't get displacement in those tissues. But so the question is if we did a block or we gave some kind of a challenge at binding potential that we know would release endogenous opiates, then we could tell what's going to the receptor and what's not actually binding to the active receptor. So subtraction kind of thing.
Mm-hmm.
Yeah.
Yeah.
That
You're
could
not going to be able to make a direct measure, Julie, but it's possible we could come up with some other correction factor based upon binding in other regions. But
Yeah,
probably outside the brain.
cell type is going to be tough. That's going to be tough.
Right.
We have our second carfentanil scan scheduled for tomorrow, so we'll have a couple animals to look at. And for those of you on the P30, we will be showing those images at our next P30 monthly meeting.
You're just going to tease us? I was going to send you an email if we could see that. You're not going to. I got the slide, but I'm not going to show it. I could.
Oh, sure. Okay. All right. Now that you're prompting me, we have a few minutes left. If you stop sharing, I'll be happy to share my screen.
Please do.
You just got to keep wanting more, Jake.
I don't. When someone asks me for something new, I immediately send a slide. I don't stop.
Well, I just got a little.
I
Wow.
think this is really interesting. I mean, I think this whole neuroimmune sort of area is, and the role of the opiate receptors on microglia and some of these immune cells is, I
It's
think
huge.
it's pretty important. Yeah.
And it's a hot area. I mean, it was one of our speakers, right, that was talking about the intersection of immune and inflammatory function and HIV and opioids and the brain barrier. It's a very rich area and a complex one, but then Explorer is going to make it cool. So, Jake, let's see, Bob.
Something just happened. It locked up.
Ah,
Am I still here? I've got some.
you're still there, but you're not sharing exactly. I mean,
Well,
you're a sharing person, but you
I
don't
think
have the
it's
slide up.
the blue jeans somehow. It's kind of locked up on me.
Probably has some kind of permissions issue.
Always. I have to wait for the next episode. Always.
Yeah, I can't. I'm sorry about that.
I'm sorry,
Yeah,
I don't know how to. I don't know how to do anything about it. Sorry, I think I'm technically the moderator because Chris Petro gave me the magic number for being able to start a session when he's not automatically here. So, but I don't know how to unlock things.
Yeah, I'm completely locked up for some reason. Sorry, guys.
No, that's all right. Well, you just whetted our appetite, I guess, for the next.
Well, I guess we will have to wait.
Well, it looks like there's several areas that got tweaked here in terms of basically areas where there's very little known. I mean, it's very interesting that they've looked in these other transmission systems, but haven't really looked as much with opioids in terms of these effects. I think that's curious. I guess you think with all the heat and light of the discovery of the endogenous opioids and the endogenous opioid receptors, and that's been going on obviously for decades now, that they would have done a lot with these seasonal things,
Yeah, I would expect there must be some study looking at the opioid system and seasonal affective disorder. I mean, that's a very, well characterized.
Yeah, seems right for that, and especially since they've made a big deal. Irwin Walecki had a whole path, and people do, on looking at buprenorphine as a kappa antagonist for treatment-resistant depression, not just seasonal, but just regular old depression. So, I think that there's it's interesting that there isn't more data looking in that direction. So maybe for one of our next journal clubs, who knows?
Actually, one other thing they haven't
I'm
addressed.
really disappointed by the nocturnal animal kind of parallel. So you can image rats because everything works in rats sometimes, but it's like they're not human biology. They don't have the same circadian rhythm as we do.
The other thing that they didn't address, which I think kind of is important, is I didn't see anything where they talk about how much daylight hours. I mean, like sunny daylight hours, because a lot of times when you talk about these seasonal adjusted disorders, they really hit hard in the Pacific Northwest because how cloudy it is there. Whereas they always say, some would claim in Philadelphia it's always sunny. So that might play into it as well.
Yeah, they didn't have any information about clouds or
Yeah,
covering
they did
versus
factor
sunshine
in
is
that.
just
They did.
Like
sun
I'd
over
be
the
interested in their
horizon. Yeah.
vitamin D levels or something like that too.
Yeah.
You know, 'cause that's supposed to be directly attributed to how much solar radiation they're exposed to, and that's more what I think the light treatment would be affecting as a more readily measured variable. But I still applaud them for taking, data they had
Oh
access to
yeah,
and trying
I
to
think
feed
it
it
was a
from that.
creative,
So
provocative thing to do, and obviously, if you started collecting data right now, it'd be a very long time before you would be able to do this in a prospective way with pet and humans. So kudos to them for looking at their existing data sets, and they must have found a way, obviously, to combine across their scanners and their platforms, because otherwise, I haven't looked at this group specifically, but they wouldn't have been able to use so much combining of data, right? So, and you would always hope to, because the data is so preciously acquired. It's great to be able to combine it when you can. Oh, look at this.
You see it?
We do.
Yes.
yay!
I finally figured it out. Okay, this is for my group meeting, but let me see if I can get the movie to go. So there's
Whoa.
the movie showing the uptake,
Wow.
and you see this accumulation in the brain,
You
but you
sure
can
do.
see accumulation in the spleen. Let me advance it a little. Here are the different brain regions.
Very beautiful, by the way.
That's a cool scanner. Very cool.
Which how is this? This is one of the older animals, Bob?
Now, this is Mason. He's relatively young. But
Gotcha.
you can see that there is uptake in this. And this is not the spinal cord. This is actually the spine
Yeah.
Huh.
in the marrow.
Yeah. Wow. That's wild.
And going back, I think PJ, I guess she didn't do the spleen, but we do see uptake in the spleen clearly.
Yeah, you can see it pretty well.
And you
Yeah.
know how he's using the DVR. CJ was doing the DVR instead of the SRTM. That's a little robust. It's more robust and easier to get right metric
see-
than what they chose to do. Makes
you
me
can
really
see-
suspect they just took it somewhere else.
Sorry, you can see spleen, pancreas, and adrenals there. So
Yeah.
not-
So I guess getting back to your question, Julie, I think we do see uptake of organs known or to have immune function. Okay, and so we might be able to look at the influence of these things on the immune system as well as the brain. We might be able to tease out neuroinflammation directly with this. We'd have to use something else. Okay.
Bob, I think those are axillary lymph nodes you can see too.
Yeah.
Ah, look at that. Yep. Oh,
Thank-
cool. Right there. Yeah.
There.
Yeah. And on the other side. Yep.
Yeah.
Cool.
Yeah.
That's really interesting.
Oh, cool. Gee,
Okay.
that's better than Netflix. Thank you very much.
Oh, you're
A little more
welcome.
expensive. The subscription is a little more expensive than Netflix.
So we will show this at the Unite P30 meeting in a couple of weeks. Okay.
That's
Cool.
nice. Well, that was
All
a
right.
real treat. This is a double treat from Darrell in terms of a very clear overview of the article with all of the caveats and a good discussion and a data treat at the very end for dessert. So probably more than I could ask for at 3 p.m.
All right.
Any
Very good.
other comments?
Good. Thank you,
Okay.
Darrell. That was
Yeah.
great.
Thank you so much, you
Thanks,
guys.
everyone.
And we'll look forward